My name is Ilan Gronau. I'm a faculty member at Herzliya Interdisciplinary Center. It's a small teaching and research institute right north of Tel Aviv. And what I focus my research on mostly is population genomics, which is pretty much kind of the field of trying to understand how population history and evolution shape present-day genome sequence variation. And one of the reasons why this is kind of interesting to do, especially in this era now, is that we have a lot of genome sequence variation data that we can actually use to try to reconstruct evolutionary history of different species, different populations. Now, this is what, like, many people in the field do, and what I like specifically to think about is ways where we can this as well. So when I talk about population history, there are kind of two aspects usually that people think about. One is the demographic history of populations, just how a population changes in size, splits, then sometimes merges due to gene flow. That's kind of the demographic history. And that describes the neutral variation along the genome. So you can think about it as, because this demographic history affects all the genome, all regions in the genome the same way, you can think of it as kind of the average effects along the genome. Where particularly interesting things you can actually do is look at differential effects, where different regions of the genome are affected by natural selection in a different way, and that shapes our variation as well. So there are these two aspects, and I'm going to talk a bit about both of them today. So that's what all population geneticists do. My kind of specific angle is to try to think of ways where we can actually use this explicit representation of genetic genealogies that sits in the middle, kind of between the population history and our genome sequence data. So what we're going to do today is we're going to start by talking about this genetic genealogy component, or what's called the ancestral recombination graph, or ARG. I'm going to devote the first third of the talk to talking about this, and then I'm going to talk about two examples of places where we used ARG inference to address interesting questions having to do with demographic history, and that I'm going to use an example I talked about in detail last year, so I'm only going to briefly summarize that. In inferring kind of odd admixture events between humans and Neanderthals that was very difficult to get without this explicit ARG representation. And the second example, in kind of the last part of the talk, is going to be an ongoing study of how we are going to use ARGs to study speciation, particularly in birds, but it's obviously a general application. So let's start with understanding what is this ancestral recombination graph and how can we actually infer it from data. So the ancestral recombination graph is pretty much your basic genealogy, right? So if you look at a genealogy, and this is you down here, and you have 2 parents and 4 grandparents and 8 great-grandparents. So it's kind of a tree branching backward in time. But if you move back in the generations, at some point, these lineages are going to start merging or coalescing, right? So it's not really a tree structure, it's more like an intricate graph structure, even when you look at the genealogy of a single individual going back in time. Now this becomes even more interesting when you look at the shared genealogy of several individuals. Here you see the shared genealogy of Alice, Bob, and Charles. So this genealogy is kind of traversing back in time in 3 different populations. And you can get from this genealogy a lot of interesting clues. So for instance, you can see that it was a population size reduction here because there is an excess of these coalescent events, right? So assuming that 2 lineages, that each lineage chooses kind of its parent independently at random from the previous generation, there's going to be more coalescences the smaller the populations are. So by looking at the coalescence rate, you can infer population size, and you can also infer kind of the splits. You can see that this population is more distantly related to these two, and they're more closely related to each other. And there is also this kind of gene flow here, right? Most coalescences between lineages from Alice's ancestry or Bob or Charles's ancestry trace back to this population back here, but there are some maybe sparse, more recent coalescences that we can use to maybe infer some gene flow between these populations. So this just shows that the genetic genealogy is very useful, has a lot of useful information about population history, and we can also, so this kind of gives you the demography aspect of that I talked about in the first slide, but you can actually get, now looking at different parts of the genome, they're going to realize different parts of this genealogy, and I'm going to show you later how we can actually use the structure of this genealogy to infer selection as well. So this doesn't have to have anything to do with genomes yet, right? This is just, you can think about it as the recorded genealogy of individuals. Say somebody was able to write it tens of thousands of generations back in time, you would get this structure. But we obviously don't have that, and we want to be able to infer this from genetic data. So where does that information come from? So you can think about now the genomes of Alice, Bob, and Charles aligned to each other, and we're focusing now on a single position in these genomes. And if you're aligning them, then each of the bases here, you have an A and a C here in Charles, so each base was inherited in a single line of lineage going back in time. So say this A in Alice's genome traces back in this line of ancestry along this genealogy that we saw in the previous slide. And the same, we can trace this A of Bob going here, and it will merge with Alice's line of ancestry here, and Charles's line of ancestry emerges up here. And this explains maybe why Charles has a different allele, because there might have been a mutation here from a C to A that gives you A in Alice and Bob and a C in Charles, right? So this is the local, what's called the local genealogy or local tree in this specific position, and it's kind of a tree embedded in this big in this big genetic genealogy. Now this tree, if you start moving along the genome, so if it's a narrow enough window, you still get the same tree, right? The same tree for all of these positions in this short window. But then as you start moving outside this window, you start encountering changes in the tree. You can see that, say, in this region, you get a slightly different tree. And the reason you get a slightly different tree is because there was an ancestral recombination event. So that's represented by these green dots here. So, for instance, at this position here, there was a recombination event where right from this spot in the genome, this green individual inherited this part of the chromosome from their, say, mother, and that led to this lineage going to the left. And left of this recombination event, they inherited that part of the chromosome from their father, right? So that single switch will lead to a completely kind of independent lineage going back in time, and now you're going to sample a different lineage up here. So left of this point, you get this lineage that coalesces at this more recent point with Charles, and here you get an early coalescence actually with Alice. So these recombination events are the events that actually change the tree as you go along the genome, and some of them change it substantially, like here we actually change the structure. This is kind of a very informative event. Some are less informative, right? It could be just that the switch changes the coalescent time, but it doesn't really change the structure of the tree. So now if you go along the genome, each part of the genome is going to be a realization of a subset of kind of a subtree that is embedded in your genetic genealogy, and hopefully, right, if you have, now we have complete genome sequence data, we can actually get some information on this entire genetic genealogy, right, if we read information from all different positions. So this is pretty much what the ARG is. It's the structure, it's a graph. It's a graphical structure that exactly describes this. It describes the local genealogies in each position and how they change. One local genealogy changes to become kind of the next local genealogy in this sequence. And together, you can kind of reconstruct the entire genetic genealogy. So an ancestral recombination graph is pretty much just a genetic genealogy of your data. So this could obviously, is extremely useful in population genetics, but most people opt to kind of circumvent this and think of summary statistics that try to kind of capture features of this without actually trying to infer the actual ancestral recombination graph. And the reason for this is that inferring the ARG is very challenging and practically impossible. So why is it very challenging? So one approach you might want to take is to kind of maybe understand what these windows are and then use each window to reconstruct a tree and then thread these trees together somehow. Yeah?
Just a quick clarification. Is the ancestral recombination graph individual black lines, all black lines, or that pale blue sort of tree-like structure behind it?
It's just the dark blue part. You can think about it's all the lines here. It's not the population history thing, yeah. So the ancestral recombination graph or the genetic genealogy, it's the same thing. They're embedded in the population history. So hopefully if you can infer the ancestral recombination graph, you pretty much have a lot of features of the population history in there that we saw on that slide with population size changes and stuff, yeah.
So the ARG is just a set of disjoint trees
They're not that's the problem. They're not disjoint. Well, as we'll talk about, you'll see it. It's more complex. So they're not disjoint, but at the end, it's a sequence of trees that are actually highly correlated. They're not independent in any way, and also the information of how you move from one to another, because you can have two trees. The fact that you know what the trees look like doesn't mean that you actually know where the recombination event happens. So that information is also part of the ARG as well. But you can think of it as a set of trees as well. But however you think about it, it's pretty much impossible to infer from the following reason. So as I said, one approach that seems like might work is that you can try and understand where these recombination events fall and then use each window to infer a tree using phylogenetic reconstruction methods that Sagi mentioned yesterday. The problem is that each of these windows is very, very short. Recombination rate is about twice as slow as the mutation rate, meaning that on average you have something like 2 mutations in each of these blocks. So even if you could tell what the blocks are, which is a complex task in itself, you don't have enough information to infer the tree here, right? So you'd have very noisy information and you wouldn't be able to stitch these together. So you have to have, if you want to be able to do something, you kind of have to pool information together jointly from all of your segments. The other challenge is that, as I said, is that these trees are highly correlated, right? This tree and this tree, they look very much the same. They're only different in this like small part where the lineages, were independent. So here they even look the same. Here they look a bit different, but again, all this part is exactly identical. So we want to be able to model this, and that again creates a complication, but there's also kind of a silver lining here, because the fact that they're heavily correlated means that there might actually be some signal here. For instance, to infer this event here, the fact that there was gene flow, as we've seen in the previous slides, from Alice's population to Bob populations, we don't just need this segment, we can use this longer segment here. All of this has evidence of this gene flow, despite the fact that the tree changed along the way. So again, there is reason to think that, there is reason for some optimism that we might be able to infer some things, but we're not going to be able to have a nice ARG to read to do that. So again, that leads to the final point, is that even if we were very, very smart and were able to pool the information in kind of an optimal way, most features of the ARG are not well supported in the data. So when you think about kind of phylogenetic terms, when you do phylogenetic inference, again, as Sagi showed you yesterday, you can kind of collapse the parts of the tree that there is no evidence of, right? And lead to, you're just going to reconstruct a non-binary tree. With ARGs, there is no really good parallel to that, because you could do some collapsing, but there are other parts where there is uncertainty that you can't really model with collapsing part of the tree. So there are combinatorial approaches to try to reconstruct the ARG, but they're not very successful. They're nice algorithms, but they weren't very useful in getting kind of useful features of the ARG out. But approaches that were successful, and this is just kind of one example of an algorithm that was able to do this, but there are other algorithms that follow a similar approach that are also successful, is a probabilistic sampling approach. So the idea is that you're not going to try to come up with one ARG that explains the data in the best way. You're just going to come up with, say, 100 or 1,000 ARGs that are kind of that reasonably fit your data, and then use the distribution of the ARGs kind of to say something informative about your population history. So the idea is, so this is a method, this is kind of the only really method slide here. I'm not going to spend much time on this, but it's a really cool method, ArgWeaver, that does this, that samples ARGs. So just kind of highlighting it. So the way they address the main challenges that we saw in the previous slide is, first, that you don't really reconstruct the ARG segment by segment, but you really sample it jointly for a very long region, 20 megabases. You just sample an ARG for that entire segment. Another thing is that the correlation between these trees is modeled using a kind of a Markov model that allows you to use kind of a lot of hidden Markov models in this sampling. This seems like it would be a great model for recombination. It turns out that the actual model of recombination is not Markovian, so this tree here is not independent of this tree once you know this tree in the middle. So there are some correlations, kind of long-term correlations, but you can get away with that. And the final thing, this is kind of the neat trick, the neat specific trick that ArgWeaver takes, is this rethreading approach. So you start with kind of an arbitrary ARG, you just guess an ARG for your data, then you choose a lineage and you look at that lineage along your trees and you take it out. So you now have an ARG on n-1 samples. And what you do is you resample this, you kind of rethread this lineage on your existing ARG with n-1 samples and given the data. So there's a very nice process. You can kind of create an HMM, a hidden Markov model, that allows you to do this threading pretty much as you were to do, you kind of resample this lineage according to an approximate posterior distribution. So now you're sampling it based on the data. So if you do this enough times, you kind of start diverging from your initial arbitrary ARG, and you start getting ARGs that really fit the data and converge to the posterior distribution of ARGs given the data. So after you've done this, say, for 100 iterations, you do it for 100 more iterations, have your 100 sampled ARGs, and then you can use statistics on these ARGs to answer cool questions about population history. So I just want to highlight Matt Rasmussen. He was a postdoc at Adam Siepel's lab, where I did my postdoc. He was the one who developed ArgWeaver. And Melissa Hubish, she's a grad student at Adam's lab. She's now continuing work on ArgWeaver. So they're the big brains. I just like using it and thinking of nice uses of it to do cool inferences. So this is the first example I'm going to show you. Is about human population history and admixture with Neanderthals. So as some of you may know, our kind of our not so deep evolutionary history of humans have us kind of split into two separate groups. our ancestors were pretty much confined to Africa, say 300,000 years ago, and kind of a separate group then evolved in the Middle East and Europe and Asia, and these were Neanderthals. So for about 500,000 years, there were two separate groups of humans that evolved in different parts of the world. And then something like 65,000 years ago, our ancestors started migrating out of Africa and encountered these populations in Europe and the Middle East and Asia. And, you know, for a long time there was this long-standing question of whether there was, so there seemed like there was chance for interaction and gene flow between these two species or populations, but there was no real evidence. And now with the advent of ancient DNA sequencing, we could actually look at Neanderthal DNA and see if we see signs of gene flow from, say, Neanderthals to present-day humans. So as probably a lot of you know, when you analyze these data, so we now have several samples kind of Neanderthals from different parts of their geographical distribution. And when you look at the data, you see a pretty striking image where there is quite a lot of gene flow. So it's not a lot in terms of percentages, but you see a lot of these recurring events of gene flow from Neanderthals and other populations, other Eurasian kind of archaic humans into modern humans as they spread into Eurasia. So just to kind of go through this image, so here you see what looks as like a very thin line is actually kind of most of human diversity is in African populations, but there's not a lot of interesting things happening here in terms of admixture with archaic humans because their ancestry was in Africa and their ancestry was in Eurasia. But when you look at all the human populations that have ancestry outside of Africa, then pretty much all of them receive gene flow from Neanderthals. This kind of represents Western Neanderthals. This is more Eastern Neanderthals. And this branch is called the Denisovan subspecies or population. It's actually, you know, specimen. We don't have a physical specimen of Denisovan. We only have a few fragments of bone. This is the kind of the famous Denisovan bone from which we have the high coverage genome. So we pretty much have just genome data. That's all our species is in this genome data, but it's divergent enough from Neanderthal so that we can call it a separate population. And it also has separate contribution into present-day humans in Southeast Asia and Melanesia that you don't see from Neanderthal. So it's a pretty kind of intricate network of gene flow. But as you see, most of these events are consistent with this image that we have of our ancestors spreading in Eurasia and meeting these populations that were established there and mating and then being some gene flow. And then for some reason or another, these archaic populations got extinct and we survived and now we carry their genes. So each of us carries about 1 to 3% of Neanderthals in our genome. But one event that's kind of curious and doesn't get a lot of attention in the media is this odd event that was found when the high coverage Neanderthal genome was sequenced in 2014. And this is gene flow from an even more early divergent population. It's called kind of a super archaic hominin that diverged something like 1 million years ago from this entire group. And it contributed some gene flow into the Denisovan genome, right? So it's very odd, and it's actually very exciting in terms of evolution, right? Because you have this very separate species, say something like Homo erectus, that contributed genes to Denisovan and possibly maybe in an indirect way to some modern day human population. So if we could kind of understand this event and maybe possible contributions to human population, it could be really, really great. So in that paper in 2014, they have a nice, question.
This might be a naive question, but all the arrows show the gene flow from Neanderthal to human. Is there none in the other direction?
We'll get to that. but none of these are in the other direction. So one reason is that all of them actually occurred after these genomes were sampled. So these genomes, these individuals died, say, 50,000 years ago. So what you see is not gene flow directly from the sample. It's always ancestral to that. There was another population that lived later, and that one contributed to humans. So you will never see, so gene flow that occurred 50,000 years ago, you're not going to see in a genome that's about 50,000 years ago. So you can't see even, so it's not that it was unidirectional, but the way we sample it, it seems unidirectional. So if we're going to, so it's possibly, hypothetically, if we were able to sample a later Neanderthal, and there are some attempts, they're not as high quality, you might be able to see some human genes there as well. But we are going to see some arrows moving in the other direction soon. So going back to this kind of curious event, so in that paper they talk about this event and kind of what features of the data support it, and there's about, you know, one paragraph in the paper and about 40 pages in the supplement talking about this arrow here. So it's a 250-page supplement, so it's not like all the supplement. They all come with this nice book with all the actual analysis. The paper itself contains very little. So there was a lot of work put in establishing this event, and so I'm not going to be able to summarize these 40 pages, but I'm going to talk about like the main signal that got their attention to this type of event, and we're going to see how to try to interrogate this signal deeply with ARGs. So what was that event? What was that signal? It goes back to the kind of the basic test of admixture that people are using when trying to see if populations are admixed. So what you do is you take 4 genome samples. One is an outgroup, that's going to just be chimpanzee, and you're going to have 3, in our case, it's going to be Denisovan, Neanderthal, and an African individual. You're going to look at these 3 genomes. So if you consider the kind of the population tree of these 3 samples, it's going to look like this, right? Denisovans are more closely related to Neanderthals, and Africans are kind of an outgroup to them, and chimpanzee an outgroup to that. And then you're going to look at sites where only one of your 3, exactly say one of your 3 genomes shares the chimpanzee allele, right? So you're going to have 3 types of sites. You're going to have a site where the African genome shares this A allele, or the Denisovan genome shares the A allele, or the Neanderthal genome shares the ancestral chimpanzee allele, right? So, and you can think about each of these site patterns as corresponding to a different local tree, right? This will be the local tree that has the same structure as your population tree. So you expect to see many more, like most of your sites, if you think about kind of a histogram among these 3 sites, then this is going to have the majority, right? Because this is going to, most of your local trees are going to look like this, but you are going to see some local trees that look like this, and that's mostly going to be because, you know, your Denisovan and Neanderthal lineages don't always coalesce in this population here, and if they are kind of freely evolving back in this population, they're both free to coalesce with the African lineage. So because you have a lot of this ancestral coalescence, most coalescences actually don't occur like in the immediate time where, you know, they can occur. They occur much earlier in time. Then you actually see quite a lot of these sites as well. The thing is, if there is no gene flow, then you expect to see these site patterns, but they're going to be pretty much in the same proportion. And these admixture tests try to see if one of them is kind of significantly in higher proportions than the other. And in this particular case, indeed, they saw that there were more sites of this ABBA configuration where the Neanderthal and the African, the present-day African, share a derived allele compared to when the Denisovan shared a derived allele with the African. So there is some asymmetry, right? You can't explain this with this kind of model. You have to have some either some connection between Neanderthals and Africans, which seemed unlikely when they saw the signal, or, again, as they hypothesize, gene flow from kind of an ancestral population that diverged here and then gave some gene flow to Denisovan. That would cause, again, the same topology where Neanderthals and Africans are more closely related than the Denisovan, which would be an outgroup to them. So the 40 pages are spent to kind of try to look at these two hypotheses and try to see which of them seems more plausible. And their conclusion was that this one was more plausible. This is less supported by the data. So we encountered this when we were actually doing our we looked at the high coverage data ourselves. We did some demography inference stuff, and we were getting pretty convincing signals of gene flow from a population ancestral to Africans into Neanderthals, actually. And at first we thought it was an obvious kind of artifact, or maybe we were not modeling this correctly or something, but we thought we had this kind of, we had this itching feeling that maybe there was something here, and maybe we can actually use these args. If we could use ArgWeaver and run it on the data, we can actually use it to try to better distinguish between these two scenarios. So that's exactly what we did. So the idea is actually pretty simple. So apart from actually being able to run ArgWeaver on the data, which wasn't entirely trivial, but once we got it running and getting reasonable args, we would pretty much, the idea was to try to look for regions where ArgWeaver would infer these types of local trees, these ABBA trees, and then interrogate these trees in different ways and try to see whether they fit more this type of event or that type of event. So what exactly did we do? So why did we think that there was kind of a distinguishing factor? This is shown by this cartoon here. So the tree in dashed is what you expect to see with no gene flow, right? Neanderthals coalesce somewhere in Denisovan. That would be like the majority BBAA tree. Now, the reason why you get this ABBA tree is if you had this super archaic introgression, it would mean that a lot of the ABBA trees have this deep ancestral coalescence with Denisovan and the root of the rest of the tree. This point gets pushed up here. Whereas if you have gene flow between African ancestors and Neanderthals, then this point here would be pulled down here. So it's a different effect leading to the same topology. So we thought if we could actually get reasonable estimates of these ages here, we could be able to tell these two events apart. So specifically, this was the test we did. So we had two present-day Africans. That gives us 4 lineages because each genome actually we have 2 haploid genomes in it. So it has kind of 4 samples in it. And we looked at the African, what we call the African subtree, and we looked for places where the Neanderthal, where one of the Neanderthal lineages coalesced within this African subtree, and the Denisovan lineage is coalesced somewhere above that point. So these are a particular type of ABBA trees. Not all ABBA trees look like this. And then we tried to see what is this age, when is this kind of African component contributed into Neanderthals, and we tried to see if there is in some of these trees where we see a support for a very recent age contribution into Neanderthals. So, we could get a histogram of ages, but we'd want something to compare it against. So what we did was pretty simple. We just flipped Neanderthals and Denisovan and did the same thing, flipped, and used that kind of as a control. We tried to see if there is kind of an interesting shift in the distributions. Okay, and this is what we get. So the red is the Neanderthal distribution of ages here, and the blue is the Denisovan distribution of ages, and it's all this age of coalescence with the African subtree. And you can see that with the Denisovan distribution, there are barely any trees where it coalesces in the past, say, 200,000 years, as inferred by ArgWeaver. Whereas with the Neanderthal genome, there is quite a lot of local subtrees that have this structure and a coalescence that's really very early. So it's not like you can think of it, the total, first you see a total excess of ABBA trees in red, right? There are more there is more kind of weight on the red bars than on the blue bars, but that's what was already seen. What we see with Arguever is that's not evenly distributed along the ages. Most of the contribution, most of the excess actually comes from trees where the Neanderthal has a recent shared ancestry with the Africans. So that would be definitely consistent with gene flow from African ancestors into Neanderthals. So what we hypothesize, trying to kind of paint that on this image here, is that there was some early migration event of, I don't know if it was really modern human. We call them early modern humans because they diverged from, kind of before all of present day modern human population diverged. So they were not necessarily, per se modern, but they were able to move out of Africa, maybe settle in the Middle East, and there is quite a lot of evidence of these kind of early settlements in the Middle East and in North Africa of early modern humans that don't quite look modern, but they didn't, it seems like they weren't able to spread, through Europe and Asia. But even in the Middle East, they would have the potential to interact with Neanderthals and bring some gene flow into Neanderthals. And that was early enough so that we can actually see this gene flow in the sequenced Neanderthals that we have. So that is our hypothesis. And in the 2 years since we published this, we see more evidence of this in kind of different analyses of Neanderthal data. But now moving back to this event, right, so we don't want to completely discount this. We want to see if there is still evidence of this very curious and interesting super-archaic admixture, and we can actually use the ARGs to do that with kind of a similar approach, but now try to record this age up here and see whether there is kind of an excess of early divergence of this Denisovan lineage from the rest of the tree. So we get the same histogram in blue for Denisovan and red for Neanderthals. Here it's actually, it's less striking and significant, but there is an excess. Well, we know there's an excess of local trees of this type, but the excess seems to focus not maybe on the most deeply diverged, but at least kind of an intermediate diverged trees. So we don't exactly know how to interpret this, but it's consistent. If we do simulations, just a second, if we do simulations, we see that this is consistent with the super-archaic admixture, but it's not as striking. Once we kind of model the effect of gene flow into Neanderthals, there is a residual effect, but it's fairly small now. Yeah?
So on the y-axis, you have a count there. I assume that's like in one of the little windows where an arg is.
It's, yeah, a number of local trees where you see this.
So is there anything special about the particular windows corresponding to the ones that make the
well, we don't know which are the ones here. We just know like this entire bar, right? We just know the ages of these coalescents. It's not like, you mean well, like places in the genome, enrichment tests. So for the other one, we actually did that. We have a good, so if we look, maybe let's look forward. So we have these events. So here it's easier to do because you can look at all these red bars and even this red bar. They're all enriched in this event, and we did some look at what genes are there. It's not a lot, so there's not a lot to do. Here you'd have to look at this entire blue bar, so there's not a lot of enrichment there. So it's actually a very tough task to do, to pinpoint the actual segments that are consistent with this super-archaic admixture, because it's all mixed in this big mix of segments. So it's an ongoing project to try to really, so if you ask me, I think that this event actually likely occurred, but it's very difficult to study because of these effects. So we have, how much time do we have? Not much, right? 10 minutes? 2 minutes? 1 minute? Half a minute? 10 secones? Okay, yeah, that'll be enough to just give you an overview. So this is going to be a completely different topic, so I'm not going. I have only 5 minutes to talk about this, so I'm just going to try to kind of raise the appetite of how to use ARGs, not just to understand events in demographic history, but actually look across the genome and look at regions where interesting things happened in terms of selection. And we do this with this kind of bigger question of how species evolve and emerge. So if you think of, two separate populations and how they become different species, then this divergent process is pretty complex. First of all, there is probably a time where, they're not completely diverged and there is some gene flow. And then, for species to really have different, for their genomes to diverge and, for the phenotypes to diverge, they're going to have some adaptations that distort the organs specific ways to create really kind of diverging genomes. And you think about it, gene flow and adaptation kind of counteract. Gene flow homogenizes the genomes of these separate species, whereas adaptation would kind of push them to diverge. And the question, there's kind of a very long-standing question in speciation genetics, in what kind of conditions would eventually, in this kind of process, this process will eventually lead to separate species, and in what conditions it will, gene flow will overtake everything, and they will stay a single species. So we try to answer this question looking at a specific model with 9 bird species, cappuccino seed eaters in the Amazon, and you can see, so it's difficult, I guess, to see an image there. These are all males. They look quite different, right? So they're easy to classify as different species, but the females look, the females all look the same, and their genomes are very, very similar. So they're very recently diverged, and they share the same, pretty much the same geographic region. So they're diverging. There's a lot of gene flow going on there. So we did demography, inference. That's less interesting. Let's get to the kind of ARG stuff. So the main question we're interested in is, if you look at, this is FST, it's a measure of population divergence. If you look at it along the genome, FST of two different populations, then you see that in most of the genome it's pretty shallow. You can't see the mouse here. This, yeah, okay. It's very shallow, but you do see these nice peaks that we kind of thought of as putative islands of speciation. This is where divergence starts, and the genomes can kind of diverge from these types of regions, and some of them are really interesting and related to why these bird species look differently. They're like feather pigmentation genes. So there seem to be very interesting things in these peaks. So what, in this field, what most people do is pretty much stop at this. They just look for these peaks where there's divergence, study what's under them, and say, wow, this is an interesting model, we learned something. But what we're trying to do is now looking at these peaks and beyond these peaks and try to interrogate them a bit more. So one thing you can do that's pretty standard is just do your standard PCA analysis of genotypes around the peaks. And here you see 5 of the population, so you definitely see that there is some differentiation going on. This blue population up here, and the orange one here, and then the rest kind of as a different cluster. But it doesn't really give you a picture of what happened here. Like, was there some adaptation, some selective sweep? Is this neutral or not? So what we're doing now is we've ran ArgWeaver on this data, and we're trying to look at the ARG and extracting useful statistics. So the only thing I can show you is just picture of the local tree around this, and just to kind of give you the idea that the ARG contains a lot of very useful information about what's going on here. So here you see these 3 populations that correspond to the 3 clusters we saw before. And what we see here is that one of them, this melanogaster, that was that was actually blue up here, this really looks like a recent adaptive sweep. You see all of the melanogaster individuals share a common ancestor very recently. And you see like a slightly weaker sweep in the Nigrurufa clade, and this other third clade is less kind of distinct. It doesn't seem very, interesting, but they are separated. These p-values are kind of p-values of population separation. So there's something non-random happening here. Some of it has to do with sweeps, and some of it is either very ancient sweeps or maybe completely neutral, we don't know. And when you kind of move along this peak, so you see pretty much the signal stays, but the clades move around. And then at some point, some of the clades dissolve until all of them dissolve. So looking at the ARG along this region gives you a lot of rich information. And that's pretty much where we're now. We're trying to use these ARGs to create, a richer picture of how these species diverge as you move along the genome. So this is Ayala. She's a postdoc in my lab doing the work with the ARGs. And Hussein, he's a grad student with Adam Siepel's also using machine learning models to examine sweeps along these genomes. So just to kind of recap what we saw that ARGs are really cool. They're very complex, but not impossible to infer. And if you know what to ask them, if you know what features they can infer with high confidence, and they can give you really cool information, and you can use it to understand population history and selection as well. Thanks to all my collaborators and funding agencies and my lab members at Herzliya. And thank you.
So we probably have time for only one question.
Silence.
Everybody wants coffee. Well, thank you a lot.
Sure. Thank you, guys.